coming. Now we will start the second session, and we again have a very good line of speakers. Chris Donnelly will be giving the first talk, and then we will have Dr. Dang giving the second talk, and if time permits, then I'm going to give a small talk. Then I would like to open the science session for your questions. If there are any broad questions that you want to ask to any speakers, any scientists, please feel free. And again, for people who are joining via the web, please type in your questions and we will try to answer as much as we can. So without further ado, I would like to invite my friend Chris to give his talk. And thank you so much for being with us today, Chris.
Thank you. Can you hear me? I'm not sure which. We good? All right. So thank you all for inviting me, Hyundai, and for the organizers. I've been to Les Turner meeting before, but this is the first time I've been able to present. So happy to share our data and excited for the opportunity. So I'm just going to get right into it. I was sort of unsure how many slides I was going to put together, so we'll just see how this goes. So I'm at the University of Pittsburgh, and we're at the LiveLikeLou Center for ALS Research. But before I actually begin my talk, I sometimes forget to do this at the end, so I wanted to get this out of the way. Most of this work has been done by a very motivated and dedicated graduate student named Jake Mann, who's here. He has a poster, and he is looking for postdoc positions actively in the Chicago area, I might add. So if you run into him, please feel free to ask him any follow-ups, but he's really kind of the brains and the hands behind a lot of the work that I'm going to show. The underlined individuals are people who have significantly helped out with this project in one way or the other, and some of this is published and some of this is not. So if you want to look up some of it, you're more than welcome to. All right. So I'll go very quickly into ALS background, but not much. Basically, as we heard before, we don't know what causes ALS for the most part. We do know that there are about 10 to 15% of cases that have a genetic component to it. And these familial cases, we've done a lot of over a number of years since the first identification of a mutation that can cause ALS in 1993, the SOD1 gene. We've identified a large number of mutations that are causative for ALS. What's kind of interesting, however, is that even within one of these genes, we may have multiple point mutations or missense mutations within it that will be causative. So each mutation could indicate a single family. So even if you're studying a single gene of ALS, there could be multiple mutations, and whether or not one mutation is doing the same as another is complicated. And so it's a very complex genetic disease. But there is something that is somewhat uniform among patients, and that's what we see unfortunately, in postmortem tissue, and this is the neuropathology of ALS. So most neurodegenerative diseases have a neuropathology, meaning if you look at the brain of people who have been inflicted with this disease or the spinal cord in ALS, you'll actually see certain abnormal protein accumulations. In ALS, this is a protein called TDP43. It's an RNA binding protein. You can have mutations in this gene. Most of them are located in this region, which will become important in a minute, but you don't have to. In fact, what this looks like is that if you have a neuron, it's not restricted to neurons, but predominantly neuronal. In a healthy neuron, this protein is within the nucleus, but in the disease, we see in a number of neurons that you see it mislocalized from the nucleus, often absent, and sometimes even aggregated into these sort of clumps or inclusions, as I'll refer to them. Someone looks like this, so this would be what you would normally expect to see, but this is what we see in ALS. We see these mislocalized TDP inclusions when they should be in this round nuclear structure. The function of TDP primarily involves RNA binding, in terms of RNA transport, RNA splicing. It regulates the transport and translation of a number of proteins. And what's interesting about this is that with the exception of patients who have mutations in SOD1 or FUS, which is only a very small percentage of ALS patients, 97% of ALS cases have this proteinopathy. So it seems to be the one commonality within the familial and sporadic, save these two subsets of the disease, that everyone has. It also seems to correlate quite well with neurodegeneration, and we've matched that, compared that to other neuropathologies, and we do see it occurs most often where neurodegeneration is most frequent. It's not unique to ALS, but it is predominant. So in ALS, there's a lot of overlap with FTD, or frontal temporal dementia. There's a significant amount of genetic overlap in that there can be mutations that can cause one or the other. But from a neuropathological perspective, you see TDP43 pathology in about 97% of cases, but you also see it in about half of patients with FTD. Another pathology that you see in ALS, which is FUS, which I'll talk about for a minute, you also see in about 9 or 10% of FTD as well. So there's definitely overlap between these diseases, both clinically as well as sort of what we see in patients. Now, what's interesting about TDP is that if we take it out of the cell and we purify it, it aggregates. So it'll undergo this process, which I'll talk about, called liquid-liquid phase separation, in which the protein, when purified, can initially form these liquid-like droplets that mature into these fibrils or aggregates. And that's typically due, what I'll show you is it's due to this region, likely, this low complexity domain or prion-like domain in the protein. Now, modeling this proteinopathy has been quite difficult, but because we know that this protein aggregates when purified, what we typically do in a lot of models is just really overload the cell with this protein, either a wild type or a mutant version of it. And eventually what happens is you will occasionally get TDP inclusions, but the disconnect here is that you have no real control over that aggregation, and a lot of times you don't see it, but the cell will still die. And also patients don't really overexpress this protein. And so this is kind of where our story begins. We wanted to develop a way in which we could control the process of this protein aggregating to understand why it happens, and more importantly, are there ways to intervene, and even more importantly, is there giving us a clue as to what's going on in the disease, as to why this is happening in the first place. So to do this, we want to develop a new model, and we're using an optogenetic approach. So optogenetic essentially means controlling cellular processes with lights, where we insert photoreceptors or light-responsive genes or protein into mammalian cells so that it can do different things. And in this case, what we're doing is photokinetic clustering. So we took a protein, in this case, from a mustard plant, Arabidopsis, in which when it's exposed to blue light, this domain of this protein called cryptochrome-2 reversibly oligomerizes. And it looks something like this if you put it in a human cell. You hit it with a quick pulse of blue light, and the protein comes together temporarily and then dissipates. So we thought that perhaps we could create an opto-TDP, which we're calling it, and use blue light to promote TDP aggregation. So could we model TDP proteinopathy in the absence of significant overexpression? We are doing some overexpression in this model, but in the absence of significant overexpression. So really studying the process of aggregation and why this protein aggregates. So you turn that volume off. Okay. So we had to do this, we actually had to, thank you very much. We had to actually create a number of LED arrays so we could do this on like a plate scanning approach. So we had a very talented student generate these LED arrays in 96 well formats, 24 well formats. We can hook it up to our microscopes, a standard microscope, stimulate our cells while we image over time. We've also done very simplistic things where we just put a LED array in our incubator and expose ourselves to light. And so what happens if you do this in HEK cells that you've transfected with our opto-TDP protein? If there's no light, on the left, you'll see a video here, where no light, you see a nuclear signal that's pretty much nuclear, but when you have light, you get this signal where the nuclear signal is lost with the light exposure. You get these cytoplasmic inclusions over time. And this is over, I want to say 16 or 8 hours. Yeah, this is over an 8-hour time imaging. When we look at these to see if they correlate with, and they show the neuropathological hallmarks of what we see in patients, these are a couple things, which is phosphorylation of this protein, positivity for P62 ubiquitin. We do see those things. So we see that our opto-TDP protein does stain positive for ubiquitin. It is P62 positive. They're phosphorylated using hyperphosphorylated antibodies for TDP. And they look somewhat similar to what you see some of the neuropathological staining in patients are as well. So we think we've recreated TDP proteinopathy. We also had to biochemically validate this. And so we did it using a number of methods. And one here we did FRAP. So this is where we have this protein, we express it in the cell, we create the inclusion, and then we bleach it. We hit it with a pulse of laser. If it's a dynamic structure, it will come back really quickly, the signal. If it's not a dynamic structure, it won't come back. And that just suggests it's immobile and it's an aggregate as what we would expect to see in patients. And indeed, what happens is when you frap these inclusions that we can form with light, they do not recover, suggesting that they are immobile. And biochemical analysis of this does show that if you take cells expressing this, exposed to light or not light, or no light, and you look at the soluble or insoluble fraction, when you add light, you push that opto-TDP into the insoluble fraction here, and this is compared to just overexpressing TDP at the same time frame. You also get cleavage, which is something we expect to see in the disease here. you get some recruitment of endogenous, which is exciting, and you also get cleavage of the endogenous as well, suggesting that maybe it's even more of a seeding effect for endogenous, so we can maybe start this process using this tool, and then the cell will take over by pulling in the normal TDP. So it looks like what we see in patients. We've also done some studies looking at size exclusion chromatography, trying to make sure that we are getting increasing the higher order assemblies of this protein. So we did this in collaboration with Jeff Brodsky at Pittsburgh, where when we add light, we see that normally our opto TDP is on these fractions here, but when we add light, you can see it's shifting to the higher fraction, meaning that they're higher molecular weight. We also see a loss of this is all soluble protein. We see a loss of the soluble protein, but we do see a shift of the endogenous to a higher molecular weight, again, suggesting that we can initiate a seeding effect. And consistent with this, if we co-express a TDP reporter with GFP and our opto TDP, which is in red, we create inclusions, we pull in that GFP reporter, again, suggesting we can pull in multiple species. And we do think that we're getting some endogenous loss of function of TDP, because if we look at some splice targets of TDP, we see that we're getting the loss of function where TDP should be suppressing this intron, as in here, but when we expose our opto TDP cells to light, we get inclusion of this exon. And so that suggests that TDP normally isn't functioning in our model. So hopefully I've convinced you that we've got an interesting model. But the question is whether or not this is toxic. And so what we did was Jake took this protein, either the photoreceptor alone, and co-transduced that using a virus into Ren neural cultures. Ren neurons are human neural-like culture cell, similar to iPSCs, but they're immortalized cells that you can differentiate into cortical-like neurons. They're very homogeneous populations, so they're nice to study for these types of imaging experiments. And we co-transduced with a synapsin-driven far red reporter, so we can visualize the cell, as well as our optoprotein, and we did it in the presence of light or not. And we did it with either a control optoprotein, so it doesn't have TDP, but it has the photoreceptor, or with our opto TDP. And so when you do this, and this is just one cell of a much larger field of view over many fields of view and many wells, we imaged, in this case, I believe it went up to 150 hours. We're going to show you 60 hours or so. Images were taken every half hour. In this cell, it's getting the photoreceptor alone. This will show you the outline of the cell in purple. You'll see that with no light, the photoreceptor is diffuse. When you add light, the photoreceptor in the lower panel, the photoreceptor does cluster. And, but there is no significant loss of the neuron. When you do this with our opto TDP protein in the same time frame, in parallel, this is all done at the same time, with no light, you'll see that the opto TDP protein remains predominantly nuclear and the neuron seems fairly healthy. But when you add light to this paradigm, what you now get is a loss of that cytoplasmic droplets that are forming, loss of that nuclear signal, these inclusions that form, and then eventually the cell does sort of die. And we don't know the mechanism of death, but we know that those cells are no longer there. And we can quantify this quite nicely, although it's very tedious, so this took a lot of man hours. And you can see that in our opto TDP cells with light, in the red line, you're getting a significant increase in cell death over time as compared to our controls. So this is a toxic process. And when we look at what's happening to that opto TDP, we can say two things. The first is that the nuclear signal is lost. So if you look at the nuclear cytoplasmic ratio at the baseline versus at the 1 hour before the cell dies, within an hour between that cell dies, we see that in the cells that are living, the nuclear signal of opto TDP is getting stronger. In the cells that are dying, that signal is being lost. The second thing that we can say is that there are two forms of structures that are forming from our opto TDP. These larger inclusions and these smaller particles. We don't know if they're droplets or not, but they're smaller particles, we're calling them. And both of these seem to be equally toxic, because if you look at from when the time they form to when the time the cell dies, there's no real difference between the two. So these are the two events that seem to happen. And when we look at the neuropathological characteristics of these inclusions or particles, sort of at this time frame when you're getting a robust cell death, but not everything is gone yet, we see that the larger inclusions are p62 and hyperphosphorylated, whereas the particles don't appear to be p62 or hyperphosphorylated yet, or they may not mature ever, frankly. We don't know, we can't really say, but at this time frame, at least in these fixed images, they're negative for those two neuropathological hallmarks. We don't know yet what it means, but it is interesting. I will also just point out that this is a cellular model, but we've been working with Uday Pandey, who spoke earlier, in developing a fly model. And so I'm not going to go into it, but I did want to mention it, that we are getting opto-TDP inclusions by using this fly model. Now, I want to just switch gears slightly and talk quickly about can we use this approach to study other neuropathologies? So I'd mentioned another ALS pathology is FUS, usually due to genetic mutations. But in FTD, that's not often due to genetic mutations. So in ALS it is, but in FTD it's not. And we wanted to know, FUS is an RNA binding protein, so we wanted to know if we could use the same approach to create FUSopathy, just like we created TDP proteinopathy. And just very quickly, I will show you that Jake went through some painstaking efforts to do stuff this, and this is sort of our paradigm using an inducible system, again, in HEK cells. But what we are able to do is get reliable FUSopathies, FUS inclusions that are light dependent. Without light, they're very nuclear. They're insoluble by FRAP. And when we do biochemical analysis, we do see a similar pattern in which in the insoluble fraction, we're getting an increase in our opto-FUS and some endogenous recruitment of endogenous FUS in our thing as well. When we look a little further at their neuropathological hallmarks, we can say there are a few things we know about FUS FTD versus FUS ALS. And that's in FTD, you see colocalization with these two proteins. And you also see that they're not methylated, whereas in ALS, it's often methylated, but not colocalizing with these proteins. And so looking at our cells and neurons, we do see that we have more of an ALS phenotype in which we're not getting colocalization, we're getting these inclusions, but we're not getting colocalization with those two TAF or EWS proteins. We are getting methylation, however. And we are not getting stress granule colocalization as well. It's also not colocalizing with endogenous TDP either, so just a few things to note. Okay. All right, so this is great. We have this interesting model, I think, of TDP proteinopathy and FUSopathy that we can control when it happens and how it happens using light and in different cell types. So the question is, why does this happen in the disease, and what can we use this tool to kind of understand better why this happens maybe in patients? And so a couple things that I will go over very quickly and very shallowly, I think, is that one of we believe the pathway to aggregation is through this process called liquid-liquid phase separation. And what this is essentially that proteins with these domains that are similar, they're called low complexity domains or intrinsically disordered regions. We believe that they have the ability to, they're using weak multivalent forces, they're able to demix from the surrounding environment, surrounding cytosol, using attractive forces within them. So the idea is that they can demix within the cell to form structures due to an attractive cue. And this allows for some cellular organization. So for example, if we look in the cell, there's a number of structures that are membraneless, that do not contain membranes like the nucleoli, but they're structures that are there, and they're always there, and you can always look at them. One of the reasons we believe these are able to form is because a lot of the proteins and RNAs within these domains contain these within these structures contain these domains that allow the components of these coacervates to interact and kind of come together. So it's a way of sort of compartmentalizing the cell so that you can allow for certain cell functions without a membrane. Now, what we think is happening in a number of neurological diseases is that this process, this biochemical, biophysical process is going wrong. And this sort of liquid-like protein RNA droplet maybe, is now becoming too much of itself. It's becoming aberrant, so we're calling it aberrant liquid-liquid phase separation. And this can be accelerated by a number of things, maybe the environment, a loss of a component that keeps the stuff soluble, the protein soluble, an ALS causing mutation. And this is sort of a hot topic in a number of neurological diseases, not just ALS. But we wanted to study this in the context of TDP. And so we know it's been hypothesized that TDP goes through this, and the idea is that normally TDP can sort of form these droplets, these protein droplets and condensates, but if they age or they become aberrant, they'll become these pathological insoluble structures. And this can be due to genetic factors, again, or environmental mutations, but the idea is you go from this liquid-like structure to a solid. And so to do this and study whether or not TDP is able to go through this, we used an approach called an opto-droplet approach that was developed by Cliff Brangwyn. It's a similar optogenetic approach, but different in the sense that we took just the LCD of TDP43, so just that domain we think that's responsible for this liquid-like condensation of this protein, and we put it on that same photoreceptor, And the idea is that if you shine a light on the cells, if this protein domain is driving this sort of phase separation, you'll see droplets form. But if it's not, you won't. And so when we did that with the TDP LCD, we certainly saw that you were able to form these reversible droplets if you just quickly initiated that. So this is just the LCD and not the full-length protein, which is what I showed you before. So that was slightly a different method. But so what's interesting here is that Jake repeated this process a number of times. So he hit the cells with light a number of times over 10-minute intervals with a single pulse of light to see what happens if you push these LCD domains together over and over and over to sort of mimic maybe some environmental factor where this might happen in the disease context. And what he found was that the more you did this, the more irreversible these droplets became. So each successive hit resulted in longer lasting protein droplets of the TDP LCD alone. And what's exciting about this from a just basic biochemical perspective is that if you took ALS causing mutations within this domain, and we took 3 of them, and you perform that same experiment, you'll see that each mutation has a different property. So that in some mutations, after a single pulse of light, you get these very large protein droplets that continue to persist. In others, it takes a bit longer, but you can quantify this quite easily, suggesting that protein mutations, ALS-causing mutations in this domain, albeit rare, likely promote this aberrant phase transition of this low-complexity domain. And consistent with this, if we just hit the LCD alone with light for prolonged periods of time, which is what we do with our full-length protein, we can get inclusions of our wild type. So you don't need just repetitive, you can just hit it with light for a long time. So we keep promoting that LCD-LCD interaction. Okay, so this does raise one really important question though, and that is, TDP is in really high concentration in the nucleus in a cell. But I also told you that if you take TDP, you put it in a test tube, it aggregates. It undergoes lipid phase separation with itself, but it also can form fibrils or aggregates. And if you frap TDP in the nucleus, it's really dynamic. So if you hit it with frap, you can see quickly it recovers. So something in the nucleus is keeping TDP from aggregating. And this is kind of what led us to think about why is TDP aggregating in the disease, or what could be contributing to that. And so what Jake did was quite an inventive experiment, I think, and all to his credit. He took the Cry2 protein with just the LCD, what I just showed you with those droplets, and he said, well, if I do that with the RNA binding domains, do the RNA binding domains undergo phase separation? So the LCD undergoes phase separation because we showed you that, but do the RNA binding domains? And what was interesting is that even though we try to physically force the RNA binding domains together, they do not stay together. They do not form protein, undergo liquid phase separation. So that led Jake to think, perhaps the RNA binding could be doing something. So, well, the first thing he actually checked was he wanted to see whether or not if he took that LCD, which does form droplets, put it onto the RNA binding domain, could that drive droplet formation of this larger construct? But what he saw was that it didn't, which led him to think, or I think, but it led him to do an experiment in which he created an RNA binding deficient mutant of the LCD and the RRM, so it's missing the N-terminal portion of the protein, but it has these 2 domains. And when he did that, however, he was able to get droplet formation. So what it's telling us is that when TDP RRMs are bound to RNA, it inhibits phase separation with itself, which is our aberrant phase separation. And just one more experiment that I think is a great one as well, is that he did this with the FUS RRMs as well. So we took the FUS RRMs and put the TDP LCD on there, and when it's bound to RNA, it does not undergo phase separation, droplet formation. But when he does it with an RNA deficient or mitigated RNA binding FUS RRM, it does. So the FUS RRM seems to work the same way. So in collaboration with Jim Shorter and Ed Gomes at the University of Pennsylvania, we wanted to test this in purified protein. So first, we took purified TDP, we formed protein droplets, so liquid phase separation, essentially testing the ability for TDP to undergo phase separation, and it does. But if you add increasing amounts of RNA, it does not. But if you did that with a TDP that has mitigated or inhibited RNA binding activity, it still does, suggesting that the RNA binding is really inhibiting this droplet formation. And similarly, we saw the same thing when you take TDP fibrils or TDP aggregates. If you did it in the presence of no RNA, they form, but in the presence of RNA, they do not. Whereas they will always form in the presence of a mutant, an RNA-binding mutant. And if you take TDP and you do an aggregation assay based off turbidity, so you have TDP, you had RNA present in this sample, you're looking at the turbidity throughout this time course, you add RNase in it halfway through, you'll see that suddenly you get increased turbidity, which is indicative of aggregation. So both biochemically and in our cells, we think we're seeing this. And if you express a RNA binding deficient TDP, you actually get these nuclear gel-like, and some of them are solid, but some of them are gel-like droplets that are p62 positive and phosphorylated. But the one thing that's missing here is that in the disease they are cytoplasmic, and so when we mutate this, so we put the TDP in the cytoplasm, we get more insoluble structures. So we do think there's an event in which their TDP is on the cytoplasmic side. Somehow we don't know how that happens yet, but we think that the RNA in the nucleus is essentially buffering TDP aggregation, and because there's a ton of RNA that's available to interact with in the nucleus. Okay, and then just to validate this finally, in our Opto system, when we did our full-length Opto TDP, we created this, we looked for RNA localization, we saw that they were devoid of RNA. When we looked in sporadic ALS and FTLD samples, we saw, or patient tissue, we saw that, that they were also devoid of RNA, which again validates what we've been finding thus far. And then just to show you with FUS, we did the same thing with the LCD of FUS, and what Jake found is that the FUS LCD does undergo phase drop, liquid-liquid phase separation, which has been shown before, but if you put the RRM of FUS on that, it inhibits the LCD from undergoing that process. But if you mutate the RRM, now it does undergo that process. So FUS seems to be acting in a very similar way as TDP, where RNA binding is aggregating it. So this is the model that we have, wherein in our Opto system, we think we have two populations of cytoplasmic TDP. So we're creating TDP artificially, we're making it, and this is our light responsive TDP protein. We think we have some in the cytoplasm that is able to bind RNA, some that is not. If you expose ourselves to blue light, the TDP bound to RNA is unable to aggregate via its low complexity domain. But if it's not bound to RNA, it actually can interact through that domain that promotes phase separation. And that in our system actually causes an Opto TDP inclusion and also recruits endogenous. But I think the exciting thing here for us was that it does open up a possibility that maybe we could reverse this if we added a substrate to interact with these RNA deficient TDPs. And so that's kind of where we're at right now. So much of what I showed you is published, not all of it. But we developed this approach called bait oligonucleotides, where oligonucleotide therapeutics are currently being used to knock down genes using RNase H mechanisms. We're suggesting that perhaps you could use oligos to bind up proteins that, if they're unbound, will aggregate in patients or in disease. And so what Jake had done was he developed two oligonucleotides. One is a non-targeting sequence that is the same composition as this one called CLIP34, which is a published, a known RNA binding target, RNA binding sequence of TDP based off its own 3' UTR. So this will target and interact with TDP with fairly high affinity. And so we performed this assay where we created in our HEK Opto TDP cells, we pre-treated with our oligos, our scrambled oligos, and then exposed to light and quantified whether or not inclusions formed. And we saw that in all of our sample with our non-targeting oligo, they did. But with our targeting oligo, although we have mostly cytoplasmic TDP, Opto TDP, we did see a significant reduction in the number of inclusions that were formed. When we did this in our neuronal system, we actually looked at survival first. And what we saw is that when we used 500 nanomolar or micromolar of pretreatment prior to our light imaging experimental paradigm I showed you earlier, we were able to increase the viability of the two that, in a dose-dependent manner, of the CLIP34 targeting with the CLIP34 targeting treatment. But with the scramble treatment, both the neurons died around the same time as they would without any oligo treatment. So essentially, we're inhibiting cellular toxicity in our Opto system using a targeting oligonucleotide at 500 and 1 micromolar treatment, pre-treatment. We also saw a reduction in the number of inclusions or particles that were formed, and we also saw sort of a tapering off of our nuclear cytoplasmic defects that we observed earlier. So that was really exciting, and sort of in parallel, a postdoc who's now a PI at Jefferson, and Jim Shorter, who was the advisor for Lingao, were working on a project where they saw similar phenomena with FUS. And so what they were seeing is that if you took a FUS and you formed FUS protein droplets, but if you did it with just buffer or in a control non-targeting RNA, it would form these protein droplets. But if you did it with a targeting RNA, it would not. Same thing with fibrillization. If you formed fibrils, they would form in a non-targeting or a buffers treatment. Sorry, this thing's going out on me. There we go. They would form in a buffer or a non-targeting oligo treatment, RNA treatment. But if you targeted with FUS targeting RNA, you actually got your fibrillization did not occur to the same extent. And so we developed an opto-FUS system, and in that opto-FUS system, we did see, and I said Jake did this, we did see that using those same oligonucleotides, we were able to inhibit the formation of opto-FUS assemblies in HEK cells in a dose-dependent manner. The Shorter lab also showed that they could reverse the formation of these things, so not just prevent them, but reverse the formation of droplets by adding in these RNAs after they formed. Hopefully you were able to see that. Just play it back. So these are just time-lapse imaging, and then you'll see they add an RNA and all of those droplets dissipate. They further showed that with purified FUS, they were able to actually disaggregate pre-existing FUS inclusions or FUS fibrils by adding in an oligonucleotide, which is really exciting because likely patients will come into a clinic with these inclusions already. And so we would like to be able to disrupt them or at least prevent them from growing. And so in the opto-TDP or opto-FUS system, Jake developed a paradigm in which he pre-generated FUS inclusions. And once they were formed, he was able to rescue those formation of this FUS assembly with targeting oligonucleotides, whereas the control had the scramble treatment or the control treatment still had those opto inclusions. So you can see here, when the inclusions were still present, with the treatment, you're able to reduce them significantly over a short period of time of a few hours, 6 hours in this case. We're still not sure the mechanism of how they're getting degraded, if it's a cellular process or if it is disaggregation in the cell, and we're looking into that. But we are excited about this potential approach. Again, we don't know if the proteinopathy is causing the disease or some other thing, but it is something worthwhile to test. And there is a few questions that I will just address, and one is that whether or not these oligonucleotides are going to disrupt the endogenous function of TDP. If you had too much, it might prevent TDP from interacting with its normal targets. And so a paper had come out earlier this year where they were trying to do just that, basically adding in these things they're calling decoy oligos to bind up splicing factors and inhibit their ability to do their job and splice proteins. And they were actually successful at doing so, but the major difference was that you needed a tandem repeat of that intronic sequence. And in our hands, we have to use different structures. We're using stem loop structures, which are not tandem repeats. And you also needed a fair number of repeats. The other thing is that most of the oligos actually were cytoplasmic. In fact, only about 23% of the oligos got into the nucleus. So even though you're treating from the outside in using either the transfection or whatever your system is, only a small percentage actually got into the nucleus. And preliminary studies, at least with FUS oligos, are showing that only a small fraction are getting into the nucleus. So we're hopeful that we're predominantly hitting cytoplasmic assemblies and not affecting FUS function or TDP function. But just to be sure, we've been working with a CFTR mini-gene assay developed by Ayuna Ayala at St. Louis University. And what she had done, this is a mini-gene assay where TDP actually splices this cystic fibrosis gene, and what it does is suppress, if it's functioning properly, it'll suppress the inclusion of a specific exon. And so you can use this to test whether or not you're messing with TDP function by introducing this into a cell and then looking at whether or not you're messing with TDP function. And so we did that. First, we wanted to show that our positive control is we knock down TDP about 50% in these cells before doing this experiment. And then what Jake had done was if TDP is not functioning, you're going to get a significant increase in this higher band as you do with an ASO or the knockdown of TDP. But in all of our treatments with our CLIP34, as well as a modified oligo that we've been playing with, it's a little bit more potent, we didn't see any real robust change in the inclusion of this exon, suggesting that the structures that we're using, at least right now, do not seem to be disrupting TDP function in our time frame and in our experiments, although a lot more work needs to be done moving forward. And this is just a representation of this difference. So right now we're trying to figure out what we would need in order to disrupt TDP function so we can avoid that one. But we do think it has to do with structure as well. So I will end there, and I'll just say that what we've hoped I've shown you is that we created these light-inducible models of TDP and FUS. We're working on some others with collaborators like Tau. We show that we've got some in vivo models that we're still working on, although it's much slower. I may be looking for a postdoc if anyone's interested, who knows rodent work. But we do know that the formation of these are toxic, and we're repeating that now with FUS. We believe that LCD is required for aberrant phase transition of these proteins, and that if you have RNA bound to the proteins, they do not undergo this aberrant phase transition, but they can undergo normal phase transition. And an example of a good pathophysiological one would be if TDP, for example, it needs to be bound to RNA in order to get to a stress granule, which we think are probably more protective than problematic, which some people think in the disease, but jury's out. And then we think that this beta oligonucleotide approach may be something to look forward to more, or look into more, as a way to inhibit TDP aggregation-mediated toxicity for TDP and FUS. And then again, we are looking into some stress granule stuff sort of on the side. We initially were looking into it much more aggressively, but our data, which I'm not showing you, some published, some unpublished, suggests that when TDP is in a stress granule, it remains soluble. So it's probably better to be in a stress granule than not be in a stress granule. So we think the RNA deficient TDP goes into this form of these inclusions, whereas the RNA bound can go into stress granules, and maybe we can reverse this process by adding a substrate. And with that, I will end for questions. I already did my thanks, but I do want to point out Julia is a neuropathologist we work with, Uday Pandey, obviously, and Jim Shorter's work for all their help with their purified protein. So, thank you.
Thank you so much. Are there any questions?
Chris, beautiful work. I think your platform is fantastic and it will allow you to address loads of interesting questions. I have two questions. The first one is more of a clarification. So when you use a full-length TDP-43 in your Opto system, does nuclear depletion of TDP-43 precede the cytoplasmic aggregation, or do these things happen at the same time?
So it's hard. We haven't been able to answer that with endogenous. We didn't have like a reporter line. We have one now, so we're kind of looking into that. But I can tell you for the Opto, in our live imaging, and maybe Jake can jump in here if I'm wrong, but for the Opto, I believe the first thing that we saw was that there was nuclear loss. So it did seem to be an early event, and I think a lot of that has to do with the fact that what we're doing is creating little seeds in the cytoplasm because we're forcing it to aggregate as it's being translated, and then that's catching TDP as it goes in and out of the nucleus.
So it never goes back into the nucleus.
Yeah, so we don't know if we break it up, we don't know if it goes back yet.
I see.
But yeah.
And my second question is, so you showed that if you pre-treat cells with these decoys or with RNA, you prevent fibrillization, you prevent aggregation. You also showed some promising data that you can also reverse that process. That was some of the last data you showed. Is there a point of no return? So can you reverse, let's say, if you stimulate, I guess I'm trying to understand whether you'll be able to reverse long-term aggregates with this process.
So a lot of those studies are being done now. We don't know, certainly with purified protein it works. There's no question there. What we are not sure about now is whether or not the oligos are, I think we can say, at least with FUS, that the oligo treatment is preventing existing inclusions from getting larger. But we don't know if they're going away 100% or if they're getting degraded through some other cellular pathway, right? So that I think is really where we're at trying to figure out, because I don't know if in a cell it's going to be different contexts, probably other things in there, whereas with purified it's obviously just TDP or FUS in your thing. So I guess the answer is I don't yet know, but that's a really good point.
Thanks.
Sorry, Chris. I am going to ask you a question that's going to require a hand-wavy answer. Do you think that in total, your work suggests an initial transcriptomic alteration as one of the first steps in disease pathogenesis. So you have these unbound, aggregation-prone RNA binding proteins, but does that suggest that one of the first actors in disease is some,
It's a great question.
unknown RNA?
Yeah. I don't know. I mean, I think what it is telling us is that, I mean, there are other things that can inhibit TDP from aggregating, like importance, for example. But I think for us, what it is suggesting is that there's possibly something that is inhibiting TDP from interacting with its targets, either by mislocalizing it or by its target not being there. Now, there are nuclear inclusions occasionally in certain patients with mutations and also in some patients, but most of them are cytoplasmic. So I think that's one of the reasons is because there's not a lot of RNA out there. So I'm wondering almost if there is a transcriptomic change that is driving TDP out of the nucleus first, and then when it releases its RNA, it can't get back in for some reason, or it aggregates for some reason. So yeah, it very well could be something like that. I think it raises more questions than answers, but at least tells us it's a sign of what might be happening. I don't know. Is that good?
Really interesting work.
Thanks.
Beautiful too. So there was some evidence that ATP levels can regulate liquid-liquid phase separation. Have you, your system would seem to be well poised to sort of manipulate the metabolic state of cells and ask whether the propensity to.
Yeah, that's a great question. So yeah, that's something that we have a graduate student who just joined. One of her things that she's interested in is looking at things like altering things like intracellular calcium levels, because TDP does act weird that way. And maybe the microenvironment, excitability, increased excitability could promote TDP to aggregate by sort of increased calcium transients in an axon. So I don't think we know at this point, but it's one of those things where looking at sort of changes intracellularly in the context of this, we could get some more answers of how it affects it, or maybe even reverses it. So yeah, it's a really good point.
Chris,
Yeah.
question here. So we know TDP is in the nucleus, is also in the cytoplasm. It goes back and forth, back and forth. Do we know the binding partners of TDP in the nucleus and the binding partners of TDP in the cytoplasm? Is it possible that it actually interacts with different set of proteins, and depending on where it is in the cell, the binding partners may be different? And because is this really the gain of toxicity or lack of function? And what do you think about that?
I think it's probably a little bit of all the above, in the sense that I think that there is a loss of function contribution. I think there's a gain of function contribution, because we certainly can kill cells by creating protein inclusions. But I don't know the other way. Now, I will say that I don't know if anyone's done like a really comprehensive comparison of, say, like axonal TDP granules versus what's in the nucleus. They've done a lot with RNA clip-seq to look at the RNA binding partners. Every study is a little different, so you take it with a grain of salt. But I do think it's intriguing. I mean, most of the time when TDP is cytoplasmic, you would assume that it's going to be formed in an RNA transport granule, trafficking an RNA or processing a microRNA or things like that. Some people say it's in stress granules, and it is, but not as much as, say, FUS. It's much harder to see, in my opinion. So yeah, I think it's a really good point, because the protein interactors will absolutely dictate, like, what are the natural inhibitors of aggregation in the cell, why it's not aggregating normally, and now suddenly it is aggregating.
Yeah, exactly.
And so it's almost like we need to be looking for those binding partners that are now missing, and why they're not in the right place at the right time. It's sort of how our view has sort of changed about the pathology.
Exactly. Thank you so much.
Yeah.
Thank you so much.